talking about cell-to-cell communication
for Parkinson's disease.
So today we're going to talk about a brief overview of cell-to-cell communication. So cell-to-cell communication is essential for all bodily processes and functions, since no processes can start without communication between cells.
This communication occurs through a pathway called signal transduction pathway. The signal transduction pathway is the series of steps or chemical reactions that brings about a specific cellular response when a molecule is attached to the receptor. We're going to now talk about how this pathway exactly works. The first step in this pathway is called reception. During reception, a ligand travels and binds with the receptor protein in the cell's membrane. It's important to remember that the binding between a ligand and a receptor is highly specific. Next, transduction occurs. In transduction, the ligand is passed down from one relay molecule to the next, often causing the signal to convert to a form that can bring about a specific cellular response. This change in form involves a phosphorylation cascade with enzyme protein kinases, which then amplifies the signal and makes the cellular response bigger. One thing to note is that protein kinases essentially turn the signal on because they add a phosphate group to the protein, while the not drawn protein phosphates turn the signal off because they remove a phosphate group. Finally, this all leads to the final step, response. In the response stage, the relay molecule from the transduction phase eventually activates a protein that triggers the cellular response. The cellular response can vary in terms of what it actually is. For example, the cellular response may be transcription or cytoplasmic activity. They could also regulate protein synthesis, usually by turning specific genes on or off in the nucleus.
It's easy to see that without the ligand, no cellular response can occur. We need that initial connection between the ligand and the receptor. The series of steps that follow don't get the signal that they need to start.
It's like dominoes. Without our finger, we can't push the first domino, which means that none of the other dominoes fall like we need them to. Basically, the response that we want doesn't occur because our finger, just like the response that is needed for the cell, doesn't occur because the ligand doesn't reach the receptor. Parkinson's disease affects the release of dopamine into the bloodstream. Before we can see that effect, let's look at how a person without Parkinson's performs the cell-to-cell communication. It starts with the release of neurotransmitters dopamine when tyrosine activates L-DOPA. The release of the neurotransmitter dopamine in the presynaptic neuron results in a signaling in the postsynaptic neuron through D1 and D2 type dopamine receptors. D1 receptors signal through G proteins to activate adenylate cyclase, causing CAMP formation and the activation of PKA. But wait, y'all are probably wondering what G proteins are. There are different components used in the reception part of the signal transduction pathways. The component used for the pathway depends on the type of communication happening between cells. The G protein or G protein coupled receptors are receptors that, like any other receptor, detect the signal and initiate the signal transduction pathway. However, what makes it different is that when the signal reaches the G protein receptor, it causes a specific type of transduction pathway, either the CAMP or phosphatidylnocidal pathway. In the case of Parkinson's, it goes through a process that forms CAMP, or cyclic adenosine monophosphate. CAMP is a second messenger created from the ATP. CAMP transfers into cells the effects of hormones like dopamine, since they cannot pass through the cell membrane. Beyond that, CAMP activates PKA, or protein kinase A, by binding to them, which starts the phosphorylation process of proteins. This ultimately leads to the cell response of transcription of specific needed proteins. Going back to the normal way, D2-type receptors block signaling by inhibiting adenylate cyclase, which allows the cell to function normally. However, for those with Parkinson's disease, it is much more different. Since mutations of various genes can lead to Parkinson's, there are multiple ways in which the release of dopamine is inhibited, which causes complications common to the disease. For this explanation, let's focus on the PINK1 gene. Let's start with mitochondria. Mitochondria are essential for cells in that they metabolize oxygen and generate energy. Oxidative stress, which is the damage caused to cells by reactive oxygen produced during oxygen metabolism, is kept at bay from PINK1. PINK1 regulates the protein TRAP1, which in turn promotes cell survival. But when PINK1 becomes mutated, the protein TRAP1 becomes affected, which causes mitochondria damage. The damage causes too much of A-synuclein, which becomes Lewy bodies, to produce, which blocks the creation of dopamine. Hence, the dopamine never gets released, which means that the receptors never get the signal in terms of controlling motor skills, causing the symptoms of Parkinson's. So, how is this disease even inherited? For years, scientists thought there was no genetic basis for Parkinson's disease. Then, in 1997, researchers at the National Institute of Health discovered that mutations in a gene called SNCA.
Well, they were common in several families with a high prevalence of Parkinson's disease, and since then they have identified a handful of other genes that are associated with Parkinson's.
For the cases where Parkinson's is due to genetics, it can either be autosomal dominant or autosomal recessive. Complicated, right? Again, it's because of the many genes that can cause Parkinson's. So, whether it's autosomal dominant or autosomal recessive depends on which gene is altered. That means that depending on which gene is altered, you either just need one Parkinson's disease-causing allele from a parent, or if you have a gene that causes Parkinson's in a autosomal recessive way, that means you need one allele from each parent. But most cases of Parkinson's are called idiopathic or sporadic, which means
that the cause is unknown, and researchers believe that the onset is due to a combination of genetic and environmental factors.
As you know, because of the illustration we gave showing how cell-to-cell communication goes wrong for Parkinson's disease, all mutations occur on genes of chromosomes that play a direct role on dopamine cell function.
More specifically, familial cases of Parkinson's disease can be caused by mutations in the LRRK2 gene, PARK7 gene, PINK1 gene, or PRKN gene, or the SNCA gene, or by alterations in genes that have not yet been identified, because scientists don't really know a lot about this disease.
These genes are found on either chromosome 2, 4, 6, or 17. Because of the mutations on genes that are directly related to dopamine cell functions, it affects how dopamine is transmitted, which means that the cell-to-cell communication, right, is also affected, leading to this disease. We're now going to talk about the symptoms of Parkinson's disease.
So first there's something called bradykinesia, which is the slowness of movement. Basically, you're slowing down and you're losing your spontaneous and voluntary movement.
Next, we have rigidity, which is the unusual stiffness in a limb or any other body part like that.
And then there's something called a resting tremor, which is an uncontrollable movement that affects a limb.
Cognitive impairment. It is a decline in the ability to multitask and or concentrate, and potentially decline in intellectual functioning.
Also, you have problems sleeping. You get the REM sleep disorder, where individuals act out their dreams. Also, you have low blood pressure when you're standing. Anyway, bye!